What's happening, guys? Aruj again, and today we're focusing on protein synthesis translation this time. Okay, so this is going to be the second half of protein synthesis. To refresh your memory, we started off with transcription, and to transcribe something, or transcription, means to write it down, and that's going to take place in the nucleus. We're going to copy our information from DNA into messenger RNA. The second half of protein synthesis, translation, to translate is to read or interpret. We're going to interpret that message, or we're going to read it, and that takes place at a ribosome found in the cytoplasm. So if we were to go through the whole process, first is transcription. We're in the nucleus. We have our enzyme RNA polymerase, that's going to unwind a strand of DNA, and it's going to make a copy of messenger RNA based on what DNA reads. Now, again, anytime there should be a thymine in RNA, it's going to be replaced with a uracil. So RNA goes down, RNA polymerase goes down DNA, makes a copy of messenger RNA. That messenger RNA now leaves the nucleus. It's going to go bye-bye. And now we start translation. Translation takes place at a ribosome in the cytoplasm. Now, here's what's going to happen. mRNA is going to go through a ribosome, and the ribosome is going to read it 3 letters at a time. And again, we call each 3 letters, or each triplet, one codon. So it's going to read AUG first. What's going to happen next is we get tRNA, which stands for transfer RNA. On the bottom of the tRNA molecule, we have what's called an anticodon. And if you compare these two together, the anticodon and the codon, you can see that they match. They complement each other. At the top of this tRNA molecule, we have one amino acid, MET. That's short for methionine. That's the name of that amino acid. If you're kind of wondering why this looks real funky, it's because it's one strand of RNA. It's a single strand, but this strand is actually bonding with itself in different locations, and it has this amino acid up top. Okay, so that's why it has this funky shape. It's one strand, but it's folded in on itself. Okay, so we have the tRNA with the anticodon. That's going to bind to the codon, and then the tRNA molecule is going to leave, and it's going to drop off that one amino acid. It's going to go through one more time. The ribosome reads another 3 letters, another codon, and that brings another amino acid. In this case, we have leucine. tRNA goes bye-bye. tRNA leaves. It reads another codon. The anticodon matches, and it brings, in this case, valine. And it continues to read, and it continues to build this protein with this amino acid sequence. Now, before I compared protein synthesis to reading a recipe, the order matters. So for example, if you're making a pizza, you have to follow the order. You can't add butter to the dough after you take the pizza out of the oven. You can't add the tomato sauce before you make the dough. Just like that has a specific sequence, so does this. The amino acids have to be in the correct order to make that same protein, okay? And we get our chain. And the very last one is going to signal for a stop sequence. So it's going to signal the ribosome to stop the production of the protein. And then we have our protein, okay? We have our amino acids. These could fold in on itself and make a protein, but it was signaled to stop. Okay, what we're going to work on in class is I'm going to give you a chart with different codons on it. This is an amino acid chart. And the way you use this is if I give you a codon, you could tell me which of the 20, there's 20 amino acids total, you could tell me which of the 20 AUG codes for using this chart. So here's how you do this. You simply start from the center and work your way outward. And it looks like this. AUG, I go to the first letter A, and I'm going to stay in this quadrant. I'm going to stay here. Okay? A, U, I go to the next level, G, that gives me methionine. All proteins start with methionine. Methionine is actually the start amino acid. It starts all proteins. And an easy way to remember this is school starts in August, AUG, and that's the abbreviation for August. Okay. Here's another one. GCG. I start in the middle, and I find G, and I stay there. I'm going to stay in this quadrant now. I stay here. I go to the next letter, C. Now I'm going to stay in this area, and then I find G, and that gives me alanine. Now, all these amino acids, they have funky names. When we're working with this, all I want you guys to do is just write the first 3 letters, abbreviate it. So alanine will be ALA. If we do UCA, what am I going to do? I start with the center and I work my way outwards. So I find U, C, A, and that gives me serine. Abbreviate that SER. UCG, UCG. Now I still have serine. I still have the same amino acid. Now if you notice, one amino acid can have several different codons, and that's actually an important concept that we're going to talk about later on this semester. Okay. UGA, UGA, that is going to signal for the cell to stop. That's a stop codon. Okay. There's also another chart that we could use. We're going to go over it briefly. I prefer this one. I prefer the circle chart, but we could also use this table. GGG, you start off in the first position here. Whoops, let me go back. You start off in the first position. You move to the second position, and then the third, and it'll look like this. G, G, now I know I'm going to be in this box right here because I'm in this column and in this row, and then I go to the third position, and that's going to give me glycine. Okay. CUU, I start in the first position, move to the second, move to the third, that gives me leucine. AAA, that's going to give me lysine right here. Okay. Okay, so a quick recap. Actually, we're going to skip that recap.